Hi, everyone. Today we are hosting Mark Nicholas from Carnegie Mellon University. Mark is a PhD student in Eric Yttri's lab in the Department of Biological Sciences at CMU. His research interests are about how the brain makes decisions and how those decisions can be executed via movements. Before that, he got trained with Dr. Betsy Murray at the NIH, studying cortical and subcortical contributions to learning, value, and decision making. He graduated from CMU with a bachelor's degree in neuroscience, where he got trained with Dr. Sandra Coleman, studying functional plasticity in the visual cortex. And Mark will be presenting his recent neuron paper with Eric today. Thank you, Mark. You're muted.
Okay, yeah, sorry about that. Thank you. Thank you for the introduction. I'm incredibly honored to be presenting today some of our recent work looking at how cortex and striatum can contribute to both skilled and unskilled actions. I'm not going to be monitoring the chat or any of the hand-raising functions, so if you have any questions, please speak up, and we can make this more of a dialogue than just a presentation. Okay, so the overarching question that we were looking to study with this work was how does, let's see if everything works. Yeah, how does motor cortex and striatum contribute?
Okay, Mark, I'm sorry, we are not seeing your screen.
Okay, that's curious.
We were seeing it, but then it changed.
Let's see, how do I share content?
Yeah, it's coming. Okay, awesome, go ahead.
All right, sorry about that. First time using Teams to present. Okay, sorry, let me just orient the screen. Okay, so the overarching question that we were looking to study, can everybody see anything? The screen advanced?
Yes, very good.
Okay, awesome. Yeah. The overarching question we were looking to study was how motor cortex and striatum contribute to skilled behaviors. And this comes from a hypothesis put forward from the field, but I like the summary from Josh Dudman and John Krakauer in a review from 2016, such that motor cortex was responsible or permissive of motor commands, and the striatum contributes a modulation of the kinematics, or a gain modulation on the vigor of movement. And if we look to the anatomy, this is a very reduced wire diagram of how cortex and striatum produce skilled motor actions. Cortex, we know, sends direct projections to these premotor nuclei of the brainstem to ultimately produce skilled motor actions. But these neurons also send projections to the striatum, which will in turn send its projections out of the basal ganglia to influence these descending motor commands. Now, when to think about how motor cortex and striatum produce skilled motor actions, we can look to the literature to see what contributions they have, and we can see that there's some conflicting hypotheses both in motor cortex and in the striatum. So if we look to the literature, work from Ben Silvestri's group back in 2015 trains rats on a temporal lever pressing task, where the rats had to press a lever, learn the duration of a specific time window, in this case 700 milliseconds, and then press the lever again to get a reward. And then over tens of thousands of trials, we can see that the distribution of the time between press intervals got closer and closer to this target of 700 milliseconds. Now, to assay how motor cortex was contributing to this behavior, they performed both physical aspiration lesions as well as ibotenic lesions of motor cortex, and then said what happens to behavior. And if we look at lesions either after a 10-day break, there's no change in behavior. After a unilateral motor cortex lesion and a 10-day break, there's no change in behavior. And then a bilateral lesion of motor cortex and a 10-day break, there's no change in behavior. This suggests that motor cortex is not required for this recently learned skilled motor action, but one of the other findings of this paper is that motor cortex was required for the learning of this skilled motor action, such that some of the conclusion was that motor cortex is required to learn the action, and then that learned skilled motor action is pushed to subcortical areas, and motor cortex is no longer required to execute this skilled motor action. So we conclude motor cortex is not required for skilled motor actions. Some conflicting data came from Adam Hantman's work in 2015, where he trained mice to do a skilled reaching task. So the mouse is head fixed, and a little sugar pellet rotates in front of the mouse. It's able to reach out, grab the pellet, and then bring it back for consumption. So the mice learn to do this over a number of days, and then they can, let's see if this works. I've lost control of my computer, sorry. Let's see. Sorry. In a subset of trials, they would inactivate motor cortex that's associated with their right paw, right? So this mouse is a righty, he reaches out with his right paw, and you can see now that the laser is on, the animal's not able to reach for the pellet. If we jump forward, you can see the mouse is kind of using its shoulder to try and reach to grasp the sugar pellet. But once the inactivation is turned off, after 4 seconds, this motor command is released and the animal is able to reach to grab for this sugar pellet. So this suggests that motor cortex is required for skilled reaching. Now, the work from Ben Sylvetsky's group saying that motor cortex wasn't required suggested that after the animal learned the task, this skilled motor action is cached and sent to subcortical areas for action selection, such that subcortical areas such as the striatum can select for a desired action. So action selection is the different neural populations, whether it be the direct or indirect pathway, are selecting for desired movements and suppressing undesired movements, respectively. And some work supporting this comes from Lux Kravitz back in 2010, where he implanted optical fibers into the striatum and paired optogenetic stimulation while the mouse was locomoting in an open field. And every time that the light turned on at these little dots, the mouse increased its locomotion, suggesting that it was selecting for this locomotion, increasing the amount of locomotion. When stimulating the opposite indirect pathway, the mouse stopped locomoting, right? So this is supporting this go, no-go hypothesis that the direct and indirect pathway are contributing to action selection. Now, an alternative, another way of looking at the striatal activity of direct and indirect pathway neurons comes from my PI's work during his postdoc, where he trained mice to do a joystick reaching task, where he paired optogenetic stimulation with different types of reach types. And when he stimulated the direct pathway neurons, he found that there was an increase. When stimulating the direct pathway neurons on the fastest third of reaches, there was an increase in reach velocity. When stimulating the indirect pathway neurons on the fastest third of reaches, there was a decrease in reach velocity. This is suggesting that there's a this is supporting the hypothesis of the no-go idea of the basal ganglia, such that you're selecting for faster reaches and inhibiting slower reaches. Now, what's interesting, when they reversed the contingency and stimulated on the slowest third of reaches, they found that when stimulating the direct pathway, they got, on the slowest third of reaches, they got slower reaches. And when stimulating on the indirect pathway, they got less slow reaches. Or put another way, faster reaches, right? So instead of an action selection hypothesis, this is more of a reinforcement, right? So when stimulating on the fastest third of reaches, the direct pathway, you're reinforcing fast reaches and getting more fast reaches. When stimulating in the indirect pathway on the slowest third of reaches, you're negatively reinforcing slow reaches, resulting in faster reaches. Now, what's important to note is that when they removed the stimulation, this behavior persisted for tens of trials afterwards, suggesting that there was some sort of plasticity that occurred, such that the neurons of the striatum were contributing this modulation or this effect on kinematics rather than the ability to reach or not. Okay, So with this hypothesis in mind, that motor cortex is responsible or permissive of motor commands, and the striatum is contributing to this vigor modulation or kinematic regulation in the striatum, and these conflicting hypotheses about the roles of motor cortex and striatum, like any good biologist, we decided to just break the circuit and see what happens, right? So we wanted to ask questions about the role of motor cortex and the role of striatum in the production of skilled motor actions, and how each area is contributing. Okay, so when talking about skilled motor actions, the behavior that I'll be talking about today, all the trained behavior will be done on these joystick tasks. So this is a paper coming from our lab from a few years ago, where we just built a joystick rig. It's largely a build manual. This is it's relatively cheap to build. The most expensive part here is the machined metal, but it's largely made from repurposed parts, right? This is a Falcon tube that's cut off to hold the joystick. This is a pen cap that holds the joystick in place. This is a repurposed sipper tube. So it's relatively cheap to build and to make multiple joystick rigs to do high throughput behavioral acquisition. So not only did we include the hardware designs, we also include some software. So what we're looking at is one of the tasks that I'll be talking about today. So in this software, we included a center outreaching task, a response time task. These are two that I'll be talking about today, but we also had, variable amplitude tasks, and then we can also extend this into decision making, looking at two-arm bandit tasks. So what we're looking at here is a mouse that's head fixed. He's got access to the joystick, and then he moves the joystick past a threshold, a learned threshold, to get a water reward. Now, I just want to check. Can you see my mouse? Yeah, okay, great. So this is not what the mouse sees. He doesn't see the screen where the joystick is relative to the reward. The only thing the mouse gets is when they move the joystick, do they get a reward, right? So this is just for the experimenter to track the animal's behavior. Now, I want to make a note about the benefit of using joysticks compared to other types of behavioral reporting, such as nose poke or lever pressing. With the joystick, we have a very high spatial temporal resolution to quantify this reaching behavior. So not only can we get when the animal makes a decision, but how the animal makes a decision, right? We can pull out different kinematics to look at the neural correlates of the types of behavior that the animal is doing. Okay, so I want to come back to this hypothesis before, sorry, I'm just going to turn on the lights. Come back to this hypothesis of motor cortex being responsible for the or permissive of motor commands, and the striatum acting on said motor commands. It's kind of a hypothesis or idea of motor cortex says what to do and the striatum says how to do it. If we look at motor cortex, is it responsible for motor commands or not? And is the striatum, is it performing some sort of action selection, or is it doing some kinematic regulation on the descending motor commands? If we remove motor cortex, we can ask this question: what happens to behavior? And then also, in any behavior that does happen post-lesion, what happens in the striatum? If the striatum is performing some sort of action selection, potentially we should see neural activity that's associated with any ongoing movement. Okay, so to jump into some of the data, to get mice to learn skilled motor actions, I trained two different cohorts of mice on either the basic joystick reaching task or a cued response task. The basic task is the same video that we saw a few slides ago, where the mouse would move the joystick past the learned threshold to get a reward. And then there's a fixed inter-trial interval. This is a self-paced task where the animal is moving the joystick at its own volition, right? So this is an intrinsically motivated joystick reaching task. In comparison to the cued response task, this is an instructed movement, right? So this is an extrinsic motivator to instruct the animal to move the joystick. So the light turns on, the mouse makes a reach, gets a reward, and then there's this variable inter-trial interval of 5 to 10 seconds. Now it's important to we want to do two different types of tasks, one being self-paced intrinsic versus an instructed reaching task, to think about the role of motor cortex, right? So if motor cortex was only responsible for one type of intrinsically motivated behavior versus maybe not a instructed type of behavior, we could tease it out with these two types of tasks. So looking at the cued response task, this is an example of a mouse that's head fixed. So the previous video, the camera was in front of the animal, but for this video, the camera is underneath him, and you can see the zipper tube here, the mouse's hands on the joystick, and then he's head fixed here. So what we're going to see are two trials where the light turns on. The animal will move the joystick to turn off the light to get a water reward. Now, mice learn to do this quite well, and they shape their behavior to with quite low latency to turn off the joystick or turn off the light to get that reward. Okay, so in terms of a timeline, I trained mice to get to this expert level of performance. This takes about a month and a half, which is around 46 sessions. And then I performed bilateral lesions, aspiration lesions of motor cortex. And then to study the function of motor cortex, we returned the mouse to the joystick task on the day of the lesion. Now, I want to make a note about this timing because putting the mouse back onto the joystick task on the day of the lesion really assays the function of motor cortex rather than any compensatory mechanisms that take place after an insult such as a lesion. If we look to human stroke or stroke literature or rodent or monkey stroke or lesion data, we know that there are compensatory mechanisms that take place within days of the intervention or insult. So we really wanted to study the function of motor cortex rather than a partially recovered brain after some recovery period. So lesion motor cortex, return them to the joystick on the day of the lesion. And then we can ask at first pass, what happens to the behavioral performance and the kinematics after lesioning motor cortex? So prior to lesion, on the basic task, mice are readily performing rewarded reaches. So this is a reward rate relative to the session, relative to the lesion, which is on day 0. And we can see that mice are performing nice, robust reaches above this reward threshold, which is just under a centimeter. So lesion motor cortex, put them back onto the joystick on day 0. We see that behavior is lost. So it suggests that motor cortex is responsible or is required for the skilled motor task, skilled motor actions. So this is the reward rate. We see that there are some rewarded trials in the first 5 days post-lesion. We're going to be chunking the analysis into different post-lesion epochs for the first 5 days. The next 5 days, we can see it's still by 0, but after 8 to 9, 10 days, there is some recovery of behavior. So initially, motor cortex is required for skilled reaching, but this behavior starts to recover. So let's take a look at the kinematic variable of interest. What we're looking at here is the full distribution of trials. For every trial, what is the reach amplitude? This is a cumulative histogram, and the colors are associated with the post-lesion epochs. What I'm plotting here are all trials, so this is the full distribution. However, when we did the stats, we were looking at session level averages to compare between pre and post 1, 2, and 3, corrected for multiple comparisons. So prior to lesion, the median reach amplitude was well above this reward threshold of 0.9, but upon moving motor cortex, the median reach amplitude was quite small, only a few millimeters in amplitude. However, as the behavior recovered, so too did the reach amplitude, right? So the reach amplitude, the median reach amplitude became just above this reward threshold at 0.9. And you can see in post 3 and post 4, we get this nice bimodal distribution of small reaches and big reaches. So as I mentioned, using a joystick task, we can get out many different kinematic variables. We can look at the other ones. So for the basic task, we can also assay the inner reach interval, which recovers to pre-lesion levels of performance by post 4. This is the amplitude, which we just showed on the last slide, which did not recover to pre-lesion levels. That's the same story with the reach speed, as well as the tortuosity, which is the distance over the displacement. So it's how much curvature there is in a reach. Interestingly, we found that duration was largely unchanged, which suggests that the motor cortex isn't required for the termination of a reach. Okay, so this is data from the basic reaching task. We also quantified the behavior in the cued response task. And the addition of a go cue, or the instruction to move the joystick, seemed not to spare the behavior, right? So the performance was lost and then recovered. This is the response time instead of the inner reach interval. It does recover by post 4, but the kinematics also were altered post lesion. So this suggests that the behavioral performance can recover. However, the kinematics are changed post-lesion, right? So the animal is adjusting the way that the animal is doing the task to recover its ability to push the joystick. Okay, so to conclude this intermediary.
Question, Mark,
Yeah.
could you go back, please?
Yeah.
So for the cute response, the duration became longer?
Yeah, I'm not quite sure why that happened. It was robust. It was in all animals that we saw out to those time points. I really don't have an answer for that, and it's interesting that the manipulation was different, or the only changes that we saw in the basic task were largely in the beginning, post lesion, whereas it's longer in the at the end of the recovery for the cued response task.
And then the speed, tortuosity, and duration, are they computed on all reaches or only successful reaches that like reach the amplitude?
These are for all reaches.
Okay.
Yeah. Yeah, one interesting note that I found was interesting was the difference in even the pre-level speed for the basic versus the cued response task, suggesting that they're cognitively different, right? So we think that, for the speed of the cued response task, it's a little bit slower, largely due to like the speed accuracy trade-off. The mouse is really waiting for that cue to reach to move the joystick, which is different than the higher speed in the basic task. But the large finding from these two different experiments is that this temporal cueing did not help them or did not recover their behavior compared to the basic self-paced task.
And one other question, is it accurate to use the word reaching here? Because it seemed like the mouse is pushing and pulling.
Yeah, you could, they're bimanual hand movements is what I've seen in the literature. It's a push or a pull. It's a omnidirectional joystick, so that it has an X and Y axis that can move. So there's some curvature to the reach that it has, right? So there is some this like teardrop shape. So it's not a reach to grasp task, but it's a move of the joystick. So.
Right, and then my last question. So after the lesion, do you see any general behavioral impairments in these mice?
So largely no. They seem to be they can locomote fine, they can eat fine, they groom fine. We'll touch on that a little bit towards the end of the talk, but they seem, apart from the inability to move the joystick, they seem relatively normal. Okay, so behavioral performance recovers post-lesion.
Can I ask a question?
Yeah.
Sorry, before you move on. About the structure of your joystick, should the mouse just retract it himself, or is there a kind of magnet or lever to just retract it?
There is actually a spring that is at the base of the joystick. The joystick itself requires 0.18 newtons to move the joystick. So it's relatively easy for them, but I think that the outward trajectory, which all of these kinematics are calculated based on, are under the control of the mouse. The retraction of the joystick is probably a combination of the mouse and the spring that's at the base of the joystick. That's a good question. All right, so as an intermediary conclusion, we can say that motor cortex is required for this goal-directed reaching. Now, to get to what's happening in the striatum, we wanted to see what is happening to neural activity in the striatum relative to reach onset in the absence of motor cortex. So if we want to go back to the two different hypotheses, whether the striatum is performing some sort of action selection, if in the absence of motor cortex, we do see some skilled motor action, right? So we can bring back this distribution of the reaches post-lesion. If we look at post-1 and post-2, there are, about 20 to 25% of the reaches in post 1 and post 2 are rewarded, right? So these are goal-directed reaches, and these are the types of reaches we can ask what's going on in the striatum. If there is some action selection that is happening in the striatum, we would expect to see that there's neural activity that's producing or occurring during these goal-directed reaches. If instead motor cortex is sending a motor command and the striatum is just listening for this motor command, we should see that there's largely no concurrent striatal activity. Okay, so how did we do this? This is acute electrophysiology where I put an electrode in every day, both before and after the lesion. And what we're looking at here is a peri-event time histogram. So each row is a neuron. The color is the z-scored firing rate. And we can see that there are neurons that fire prior to reach onset, mostly during the reach onset, and then some. So these are all increasing their their activity, and some are decreasing their reach activity, or excuse me, the neural activity. So these are reaches that are above the reward threshold. So we can see that there's some neural activity that's out here associated with the consumption of the reward. So this is all pre-lesion. And when we lesion motor cortex, put them back on the joystick on the day of lesion, we see that the neural activity is absent. Right. So in the first 5 days post-lesion, there's no reach-aligned activity. This is the same case for post 2, as well as for post 3 and post 4. So as the animal does recover some behavior around post 3 and post 4, there's some neural activity. However, it is not comparable to the pre-lesion response profile seen here. So as I mentioned, these are for reaches that are above the reward threshold, and we can quantify these as large reaches. And we can also look at smaller reaches, right? So these are reaches that are under 0.5 centimeter, and we see that there's a nice transient reach-aligned response profile for smaller reaches, but it's much less in comparison to the large reaches, right? So this is a known finding in the field that neural activity in the striatum scales with the vigor of the movement. And we wanted to ask was, for what neural activity we see from post 1 to post 4 for big reaches versus small reaches, is this kinematic relationship still conserved, right? So for smaller reaches, we do see that there's actually an increase in reach-related activity. We wanted to see for what neural activity that we do see from post 1 to 4 in big reaches and small reaches, does this kinematic scaling of neural activity, is it still intact post-lesion? So to answer this
question, Mark, so
yeah.
These are neuropixels recording. What is your
this is recording with a Cambridge Neurotech electrode that has 64 channels, I believe.
And it's chronic?
No, it's an acute electrode. So it's implanted each day prior to the animal's behavior.
And you could do it for so many days?
After removing motor cortex, it was a lot easier to access the striatum. Yes.
Interesting, because for acute, I've heard like more than 4 times you can't really do it.
So we found that there was no changes in the intrinsic properties of the striatum. So this is, at most 23 to 25 insertions, and we're assaying, largely the same area, spanning maybe 0.5 millimeter to 1 millimeter around where was caudal forelimb area. So there's still healthy tissue that is there in the absence of motor cortex. I'll show some videos at the end, then you'll see that the mouse has a little silicone cap on its head that allows you to have access every day.
Yeah, and I can see that the number of neurons kind of decreases, perhaps, with the scale bar.
I didn't quite assay the change in number of neurons, but yeah.
Good.
Yeah. So so we wanted to ask the question of how, or if the kinematic relationship between neurons and the kinematic output, the scaling of neural activity with respect to the vigor, was conserved post-lesion. So to do this, we quantified a amplitude modulation index, where we essentially took the amount of neural activity for a big reach, and the amount of activity for a small reach and took the difference from them, right? So if a neuron was equally tuned for a big reach and a small reach, it would have an amplitude modulation of 0. So pre-lesion, we can see that this distribution, the mean distribution, is pushed to the right, probably around 2.5. Post-lesion, the amplitude modulation was lost, right? So post-lesion 1 through 4, the amplitude modulation was hugging 0, suggesting that this kinematic, the scaling of neural activity with movement vigor was lost and did not recover. So this is at the individual neuron level. If we take the session level average and quantify it as a function of the mean session amplitude, we can see that post-lesion, the neurons never recover this scaling of neural activity relative to movement vigor. So all the data I've been talking about thus far has been in putative spiny projection neurons, the major cell type of the striatum. We also quantified this for fast spiking interneurons of the striatum, although a very small proportion of the neurons compared to SPNs. We found the same results in the FSIs. There's a loss of reach-aligned neural activity post-lesion, and the kinematic vigor encoding was also lost. So to get to the question of if the striatum is still healthy, one, because we removed one of its major inputs, the motor cortex, but also because I was inserting an electrode every day, we looked at the baseline firing rate and the neuron's variability using neural activity where the animal wasn't doing things. So we took out all the time where the mouse was moving the joystick, the light was on, or reward consumption, and just looked at the baseline properties of the firing rate for SPNs and FSIs, as well as the variability in the firing rate. And we can see that between pre and post lesion, there's no change in the baseline firing rate, nor in the Fano factor, suggesting that the health of the striatum was intact with the removal of motor cortex and the repeated electrode penetrations. So this kind of gets at the idea that the intrinsic properties of the striatum are not coming from motor cortex, either inherited from somewhere else or local to the striatum itself. Okay, so back to the neural activity, we see that there's a loss of reach-aligned activity post-lesion. And all of the analyses that I've showed you thus far have been at the individual neuron level. And potentially, as the animal is doing this task and recovering some behavior, potentially there's some information about the joystick position or the selection of an action at the population level, right? So potentially at the population level, the neurons of the striatum could produce these joystick movements. So to try and figure out at the population level if there's some reach-related information still there, we built a feed-forward neural network to try and decode the joystick position from the spiking activity in the striatum. So using both SPNs and FSIs, can we use their activity to predict where the joystick is? So this is an example of 5 concatenated reaches from pre-lesion, and using the neural activity in the striatum, we can readily decode where the joystick is, right? So seen in this dashed line, we have very good decoding of where the joystick position is. However, this is lost post-lesion, right? So post 2, 3, and 4, we were unable to use the striatal activity to predict where the joystick is. Now, these are 4 example reach traces with 5 or 6 reaches each, but they actually come from the median of this distribution where we ran the model a number of different times to control for, one, the different types of neurons. So each run that we had, we'd subsample 10 neurons, and then we would subsample the number of reaches to the same number of reaches found in post 2. So that way we had an equal comparison of reaches and neurons to try and allow for this model to predict where the joystick was, and from post-lesion, the model performance never recovered to the pre-lesion level of decoding. So we can conclude that striatal reach-related activity is no longer predictive of the joystick position. So to conclude, it appears that striatal reach-related activity requires input from M1. The vigor of this encoding in the striatum is also lost and does not recover, and we can no longer predict where the joystick is using striatal activity. Now, one question that is important to address is that when lesioning motor cortex, it's quite traumatic. We're removing motor cortex, vacuuming out the tissue bilaterally, and potentially this behavior is a function due to the lesion itself, right? If we remove motor cortex, potentially the animal isn't doing the joystick task, or the striatum isn't functioning correctly because we removed part of the brain. It's a phenomenon known as dieschesis, right? So if there's an insult to one part of the brain, there's this global trauma that affects behavior and neural activity. So to address this concern, I trained another cohort of mice on the basic joystick task and then lesioned an offsite target. We went with posterior parietal cortex, which is an area that's associated with motor control in some aspects, but is far away enough from motor cortex to test this hypothesis. So train mice on the joystick task, lesion posterior parietal cortex, put them back on the day of the lesion. We see that there's no change in behavior. There's no change in the kinematics of the reaching. There's no change in the reach-aligned activity in the striatum. And there's no change in the kinematic encoding of vigor post-lesion. So this suggests that the results we see following motor cortex lesion can be credited to the function of motor cortex rather than the lesion itself. So with these results in mind,
Mark, could you please go back?
Of course.
So -3 to -1, these are pre-lesion?
Pre-lesion days, yeah, so 3 days prior to lesion and then the 3 days after the lesion. We decided to only do 6 days of behavior because we saw no changes or 6 days of neural and behavior because we saw no changes in the behavior. So we, instead of going out 20 some days, it was when the behavior didn't change and the kinematics didn't change, we were like, okay, I think this is a substantial negative control to suggest that the lesion itself wasn't what was causing our results with the M1 lesions.
Right. Well, in your speed, it seems like things get slow and then recover. Same with the amplitude, like panels FG.
I'm sorry?
So it seems like if you compare red with blue in panels F and G, it seems like we have a reduction of amplitude and speed, and then it recovers.
Potentially. Again, so this is all the trials in the cumulative histogram, but the stats are done at the session level average. So some variability might be seen in these distributions, but at the session level, statistically, we can't compare, or they're not different from each other.
I've got a question about your model that was predicting the trajectory.
Yep.
So you trained the model on the data for pre, and then you run it for the post ones?
Each model was run independently, right? So we had the same parameters, the same architecture of the model. We were just asking how well it could pull out the joystick position, and then we were comparing the performance. So we weren't training on pre and then testing on post. This was really just a test of if this same model architecture across all sessions could be used to find any reach-related information.
So it was trained on both pre and post data, but
also tested on pre and post data. So for every post-lesion epoch, it was I think it was 70% for training and then 15 for testing and validation within pre, post 1, post 2, post 3, post 4.
Okay, thanks.
Good question. Okay, so yeah, we can conclude that lesions to motor cortex are, or the lesion itself wasn't causing the behavior, rather as the function of motor cortex. So from these data, we can conclude that the striatum, in the absence of motor cortex, cannot produce this goal-directed skilled motor action. So all the data that I've been talking about today thus far has been in skilled behaviors. This is a previously learned skilled reaching task. What about unskilled behaviors? Now, I want to go back to the literature a little bit to look at the differences and the contribution of the motor cortex between recently learned skilled motor actions versus innate behaviors. So back from the Adam Hantman's work, they found that in a licking task, right? So instead of reaching to grasp, the animal could lick to receive the pellet. And during grooming, right, so the laser is on here and activating motor cortex, showing that in the absence of motor cortex, the animal can still do these innate evolutionarily conserved behaviors, such as eating or grooming. Right, so this suggests that the contribution of motor cortex might be specific to a specific context or a recently learned skilled motor action. Looking at other work from Sylvia Arbour, where they trained mice on this virtual reality task, where they have to locomote down this hallway to get to this little blue dot at the end of the hallway, which is a water reward. You see, they'll turn left, they'll turn right to try and update their trajectory to get to the reward. And what they found was during locomoting this natural corridor, they find that when turning off motor cortex, the degree of turning is not unaffected, right? So on control trials, they can quantify the amount of turning that they have, right? So the colors associated with the degrees per second. And when they turn off motor cortex, they see that there's no change in this turning, right? Suggesting that general locomotion isn't requiring motor cortex. However, because this is a virtual reality task, they have the ability to manipulate the world on the animal, right? So you can see that the world flips. There's a visual offset that the animal has to adjust its behavior. They're inducing the animal to turn, right? So this is a context-based manipulation where the animal needs to update its trajectory. And we can see that on just over a majority of the trials, when turning off motor cortex, the animal is unable to update its trajectory, right? So the extent of turning is inhibited when they turn off motor cortex, suggesting that motor cortex is required for this contextual-based updating. So this was something interesting that we saw in our own data, where on the day of the lesion, during postoperative care, we saw that animals would freeze when having to update a context, whether that be walking across the bench top and trying to hit to my, climb into my hand, they would freeze. Whether it was walking across the bench top, they'd get to a pencil, they would freeze. So to assay this, we put mice in this very complex, highly complex T-maze, where it's unrewarded. The animal just wanted to locomote, to explore. We saw that on the day of the lesion, the animal is locomoting fine, but gets to this junction and freezes. So you can see the video is still going. You can see that his ribs are moving a little bit, but the animal is frozen in its step, right? This is a freezing of gait-like behavior. He turns to the right and then locomotes down the hallway. So what's interesting, this is locomotion is conserved. However, when needing to update, the animal freezes. We made it slightly harder for the mouse, and we put a little block in the arena. He's unable to locomote around this. He freezes here. In a few, he's going to locomote past it, freezes again, and then locomotes down the hallway. I think he actually turns left. So that's an interesting finding. However, we also saw that this is within hours of the surgery, the animal's still able to groom naturally. So he can walk, what I didn't show you, but they can eat, they can groom normally post-lesion. But they do show this freezing of gait-like behavior at the junction of the T-maze. One thing to note, you can see this silicone hat that's in the head cap, which is used for access to the tissue every day. Okay, so to quantify this freezing behavior, we took this block out and just gave them the normal T-maze, and then we quantified the amount of freezing, or the amount of time spent in each position of the maze, right? So we split up the T-maze into equal parts, and then said how much time do they spend in each position of the T-maze. And this data was compared to a control group that are unoperated on. They were being used on other experiments in the lab. And you can see for pre-lesion, they spend equal amount of time at the junction of the T-maze. Upon removing motor cortex, the amount of time spent at this junction drastically increases, right? So this is about 2 minutes worth of freezing on average, almost up to 2 minutes of time that they spent at this junction in the first 5 days post-lesion. As the animal starts to recover and post-2, there's still some differences in some of the T positions, but this behavior is fully recovered by 10 to 14 days post-lesion. So this suggests that motor cortex is required for some context-based updating, right? So motor cortex is when required to update, whether it's turning left, turning right, navigating over a block, the motor cortex is required to update the context. Now, in the last little bit of the talk, I want to talk about next questions and ongoing work that we're doing in the lab to explore these same questions. Instead of using a lesion, we want to look at individual cell types, right? So can we do cell type specific lesions using caspase to eliminate PT or IT neurons, pyramidal tract or enterocephalic neurons of motor cortex to see what happens to skilled motor activity and what happens to striatal activity in the absence of these cortical striatal projection neurons? And then interesting finding with the freezing of gait, we want to try and dissect this mechanism, right? So can we explore the neural activity of downstream nuclei, whether it be the striatum, the STN, or the brainstem nuclei, to see what's going on in these areas as the animal is freezing and then recovering from the freezing, as well as from immediately post lesion and then through recovery. And then, so again, this is a blunt lesion approach with aspiration lesions. We want to translate it into an optogenetic model, right? Can we silence different neurons of M1, whether it be all neurons, PT or IT, and see if we can try and dissect this phenotype of freezing of gait, right? To my knowledge, there's not many, or if any, rodent models of freezing of gait that is a common symptoms in Parkinson's. So these are some of the questions that we're interested. Some of the other work that's going on in the lab is looking at cortex and striatum during trial and error learning, right? So I talked about the hypothesis at the beginning that motor cortex is required or responsible or permissive of motor commands, and the striatum's contributing to a gain modulation, right? So I think that our data suggests that action selection isn't happening, but to really get at that gain modulation, that kinematic regulation, we want mice to do a skilled motor action, but then change its skilled motor action. So we can explore individual neurons, whether PT, IT, DSPNs, and ISPNs, and how they contribute to updating of a skilled motor action. We're using different regressive models and concurrent recordings of cortex and striatum to try and identify the contributions of these different neurons to updating the skilled motor action. Okay, so with that, I want to thank the members of the Yttri lab. This is my website, my email. I'm on Blue Sky. If we want to try and make that that community come back, reach out. And I want to thank everybody who attended today for your time. And with that, I will take any questions and put this slide back.
Thank you, Mark. That was great. I do have several questions, but do we have any questions from the audience? I can start maybe. So, okay, so number one question, I want to understand the discrepancy between Bence's work and Adam's work. What are your thoughts on that?
So Bence's work is I think they tried to design dexterity out of that task. The role of motor cortex in a skilled motor action that is a temporal lever pressing task, they tried to design dexterity out of it. So the behavior of reaching to press the lever, wait 700 milliseconds, then press the lever again. I think the beauty of that work was largely the stereotypy in the nature of the action. So every behavior, every mouse solve that 700 milliseconds differently, whether it was stand up, scratch their left ear, press the button, scratch their left ear for 700 seconds, or touch it again, or some would do something else. These are examples. I'm not sure if they all found their own unique stereotype way of solving that problem. So that's one. The behavior is quite different.
So you think that having a one-dimensional lever versus having a spatially specific target plays a key role here, like in your joystick, because they have to go for a specific target, right? It's not just, did I get that right? Is there a target?
In our task, it's just moving past a threshold.
So it could be in any direction.
Any direction, yeah.
Why do you not call that stereotypic? Like, because that is also just pushing.
Yeah, so I will say that pre and post lesion, the direction did not change, but there was an increase in variability in the movements. So if we're thinking about the idea that the striatum is associated with the kinematic regulation of ongoing movement. If we look at the model performance, right, we lose that vigor or that kinematic information in the striatum at the population level. We see that SPNs and FSIs, that vigor modulation is lost. So we looked at the coefficient of dispersion, right? So how variable are the reach amplitudes from pre to post 1, 2, 3, and 4? And we found that between at post 4 there was an increase in this variability of the joystick movements. So pre-lesion, there is quite nice stereotypy to the movements, but post-lesion, there's an increase in this variability of the movements itself, suggesting that with the absence of motor cortex and the striatum is offline, unable to modulate this ongoing motor control, there's an increase in this variability. So between Bensie's work and this work, I think that it's asking two different types of questions. And another important variable that's quite pertinent to this is the amount of training. So they to get to their expert level of performance, there are tens of thousands of trials into somewhat of an overtrained state. And there's some literature from Komiyama suggesting that at a highly trained level of performance, there's a disengagement of motor cortex, right? So they found that on a skilled reaching task, inactivating motor cortex didn't affect the behavior, suggesting that at some crystallized state, the motor cortex might not be as important. So in terms of tens of thousands of trials in Bensie's work versus our work, we had, I think it was at max 6,000 to 7,000 trials prior to the lesion. So there's, I'd say our mice are expert, but not highly overtrained.
I see. I see. I guess that actually plays a big role here for why your results are different from Benson. And between Benson and Adam, I guess the key is in the directional reach. It's a more complex movement than
it's definitely a more complex movement for Bensie's work, whereas Adam's work seems to be a simple reach to grasp to collect a pellet. It is a skill. They do take time to learn that task. Mice are not readily normally head fixed and reaching for their food that way, so they do get better over time. But it's again also, I think, a function of the amount of training that they have, where ongoing inactivation of motor cortex at this expert but not overtrained level is a key difference.
Right. And then my other question is about this striatum. So you showed us that after motor cortex lesion, striatal encoding of movement, you say vigor. I really don't like this word means kinematics. What do we mean by vigor? Is it just kinematics?
It is kinematics. In the field, it's been described as how much ability to explain using kinematics the behavior is. So in a joystick task, it'd be how fast, how far, and the duration of the movement. In some decision-making contexts, the vigor might be the reaction time or the saccade velocity. These are all descriptions of how the animal or how the decision or action is being executed.
Okay, so the movement kinematics, the striatal activity after motor cortex lesion is no longer predictive of movement kinematics. That was your main finding. And with motor cortex lesion, even after many days, when motor, despite behavioral recovery, so the behavior does recover. However, striatum is kind of now dissociated from
it's lagging. Yeah,
exactly. So then, what's now the hypothesis here? What do you think is happening? Is it that so is the idea that the striatum was receiving this information from the motor cortex and now that's gone and that's why it cannot do it, and so there is a third area here that
so no, 20 days post lesion, the animal's doing the task. He's figured out how to do it again. But the striatum is lagging and there's no reach related information in the striatum. And I think that's a function of the timing that we're looking at. If we look to other stroke or lesion literature, it suggests that in the after lesioning or stroking, the motor cortex, perilesional cortex will take over the function. John Krakauer's got some nice work looking at this. Karnesh Ganguly's got some beautiful work looking at perilesional cortex, and they see that after training and after recovery of behavior, it's really the perilesional cortex and the striatum will link up in terms of that recovery. So in terms of our timeline, 20 days post lesion, as the behavior recovers, I believe that the striatal activity is just lagging, right? So if we went out another 20 days post lesion, potentially we would see that rewiring from perilesional cortex to the striatum and an increase in that vigor encoding in the striatum as well.
Okay, interesting. And then my last question here is about the last part of your talk, where you quantified the different types of behavior after lesion in the motor cortex.
Mm-hmm.
So you showed us that Adam did not see any of these innate behaviors being impaired, and
Right.
you kind of saw the same thing, but because raising at a gait, well, I don't know if that counts as an innate or not, but a more complex kind of behavior, which involves some sort of decision making. Am I going this way, that way? How do I avoid this this block? That is impaired. And then and this never recovers. Did you show us this, or?
So they do recover. So to quantify this, we just looked at how much time they spent in the T, right? So if we break up the position of the maze, right? So this is a normal pre-lesion mouse. We can break up the T into equally spaced bins and then say, okay, how much time they spend in each one of these little divisions, right? So, how much time do they spend in division 9 and 10 versus the other places along the T maze, right? So this junction here is the position 9, and pre-lesion we see that there's no difference between unoperated controls and the pre-lesion group, but post-lesion we quantified and we see that there's close to 2 minutes worth of freezing at the junction of this T maze. And then by post 3, it's no longer different than these unoperated controls.
Okay, and then so was the idea here to make resemblance with the reaching? What?
Less of the comparison of reaching, but this is a untrained, naturalistic, unskilled behavior. Right, we're just locomoting in an open in a T maze that's unrewarded, and when needing to update, turn left, turn right, turn around, somewhere motor cortex or somewhere the brain says turn left or turn right, but in the absence of motor cortex, the mouse can't do it. Right, so there's some updating or context switching of motor state, whether it's locomotion versus turn, that requires motor cortex, and at least initially in the first 5 days post lesion, with motor cortex offline, there's this profound freezing at that junction where the mouse needs to update. So in comparing a skilled task versus a naturalistic, walking, untrained task, there's deficits that we see in the behavioral performance for the skilled task, as well as in this context switching, this turn left, turn right.
And then you had striatum recordings here too?
We didn't record in the striatum here. That's one of the next steps that would be quite interesting to look at. Is it different as a function of in natural locomotion or natural behavior?
Awesome. We also have a joystick task in our lab, and we're very much interested in similar types of questions, mostly focused on the timing because we have sequential movements. Mm-hmm. And I would like to talk to you more about that, but I want to make sure we give chance to others. Are there any questions before we start recording and then have a more casual conversation?
That'd be great, yeah.
Sure. Okay. Thanks a lot, Mark. I'm going to stop recording now so this doesn't get too long. Let me do this. Again, thanks so much for your great talk.
Thank you.